Did you know that your childhood experiences can alter your brain's biology? Early child development sets the foundation for lifelong learning, behavior, and health. The experiences children have in early childhood can determine a child's capacity to get along with others and to respond to daily stresses and challenges. A wealth of scientific research shows the devastating effect maltreatment can have on children. Childhood maltreatment is estimated to affect up to 30% of children worldwide. The distress and upheaval is emotionally and physically overwhelming in the short term, but such a trauma is also connected to long-term biological changes that put them at risk for psychological and medical problems throughout their lives. For example, many maltreated children have interpersonal communication problems that persist later in life, with negative consequences for their social relationships, including those with friends, partners, and families. The COVID-19 pandemic creates additional concerns related to child maltreatment and heightens risk factors that existed before. Families and communities are in much worsened states of poverty, unemployment, and food insecurity. Losses and grief are affecting mental health. Social distancing and safety concerns are affecting in-person child protection work, and more. So what does this mean for us? Well, what if I told you that the decisions your ancestors made decades ago could be affecting you today? Kinda hard to believe, right? This could actually be true, because we now know that our experiences affect gene expression by changing which sections of DNA are active and which sections are inactive. Like many other childhood experiences, trauma can leave a chemical mark on a person's genes, which can then be passed down to future generations. This mark doesn't cause a genetic mutation, but it does alter the mechanism by which the gene is expressed. This alteration is not genetic, but epigenetic. Okay, so now we know that childhood trauma can alter the expression of your genes, but that's just the tip of the iceberg. Did you know that childhood maltreatment can also affect the physical development of a human brain? A recent study explores just how alterations of brain structures can happen through epigenetic changes triggered by childhood maltreatment. In this study, epigenetic data was first collected from a sample of 44 children who had experienced maltreatment, whether in the form of physical, emotional, sexual abuse, and/or neglect early in their life. The results were then compared to a demographically similar control group of 41 children with no history of maltreatment. Specifically, researchers wanted to explore the role of childhood experience on DNA methylation patterns of the oxytocin receptor gene. Whoa, that's a lot to unpack. Let's first zoom in on the oxytocin receptor, which is impaired in many children with childhood maltreatment and is suggested to play an important role in regulating social attachment behavior in various species. Oxytocin is a hormone that has an important role in many human behaviors, including recognition, trust, romantic attachment, and mother-infant bonding. The effect of this love hormone on the brain is complex and not fully understood, but its implication in many psychological disorders makes it incredibly important to understand just how epigenetic changes can lead to atypical oxytocin secretion patterns. In this particular study, researchers found that compared to the control group, children who have experienced maltreatment had higher methylation levels in the oxytocin receptor gene. But what does this mean exactly? To understand this, let's explore DNA methylation in more detail. Epigenetic changes can occur through various mechanisms, in this case DNA methylation, but they can also occur through histone modification and the activity of non-coding RNAs. So how does the addition of methyl groups silence a gene? The majority of DNA methylation occurs on cytosines that precede a guanine nucleotide, which are also known as CpG sites. This is carried out by a family of enzymes called DNA methyltransferases. More specifically, DNA methyltransferases covalently add a methyl group to the 5-carbon of the cytosine ring, resulting in 5-methylcytosine. During transcription, RNA polymerase makes its way down DNA but is blocked by the methyl groups. As a result, the gene cannot be transcribed, and the gene is effectively silenced. Researchers in this study observed that the oxytocin receptor had significantly higher methylation levels at one particular CpG site, CpG56. DNA methylation at this CpG site was found to lead to decreased expression of the oxytocin receptor, greatly impacting oxytocin signaling as a result. So can these epigenetic changes actually impact the physical structure of the human brain? In order to answer this question, researchers in this study compared structural neuroimaging data collected using MRIs to measure the changes in gray matter of the brain. Gray matter is mainly composed of neuronal cell bodies and unmyelinated axons, and undergoes development and growth throughout childhood and adolescence. They found that the degree of CpG56 methylation was negatively correlated with the gray matter volume in the left orbitofrontal cortex, meaning that the gray matter volume in this region of the brain was significantly reduced in maltreated children. This actually makes a lot of sense, because the left orbitofrontal cortex is a region that is highly relevant to emotion regulation, and it has been suggested that reduced gray matter volumes in this region is highly correlated with insecure attachment styles, higher levels of psychiatric symptoms, and higher numbers of total behavior problems. Altogether, these findings showed for the first time that oxytocin receptor hypermethylation in children is associated with difficulties in interpersonal relationships by mediating the structural alteration on the atypical brain development. But there are still some problems with this study. One being a relatively small sample size, meaning that it's not representative of the whole population of maltreated children. Another problem is that it's difficult to consider the difference in impact depending on the specific type of childhood maltreatment. In addition, although saliva methylation greatly resembles methylation patterns observed in brain tissues, cellular heterogeneity in saliva DNA can still act as a confounding factor. And DNA methylation other than that of the oxytocin receptor gene could also be involved. Nevertheless, it's still crazy to think that your childhood experiences can literally change the shape of your brain. Now that's some pretty convincing evidence for the epigenetic argument in the field of mental health. But until more studies are done on the intergenerational effects of trauma, we cannot be certain of all the possible implications. Could the current COVID-19 pandemic and the extreme stress it is causing the world at large cause epigenetic changes to offspring? It's possible, but only time will tell.